Big data analytics, enabling the insights from the whole patient population to a sample. This is a companion presentation to my Big Data Epidemiology presentation, and this builds on the Big Data Epidemiology presentation and also fills in some gaps. And this was originally delivered in sometime in 2014, and I'm transcribing this in October 2021 to get the message across. And as you see, the title is about getting insight from the whole patient population to a sample so that we can actually make a perfect sample. In general, what we do is the reverse, and we take samples and then make measurements so that we conduct studies on humans, and then we make inferences about the whole population. And we hope them to be reproducible. Sometimes these samples are systematically taken or using very many methods that have been established. Sometimes they are ad hocly sized. Sometimes they are actually properly sized using some approximations of sample size calculations. Most of them are approximations rather than any scientific theory. And here the situation in 2021 has changed because in most countries that significant proportion of the patient population is available electronically. And in some countries, the whole population is available electronically for a long period of time. And increasingly, the rest of the countries are catching up. So this mythical population is with us at the moment. So here is an attempt, as I described in the epidemiology presentation, that if we build a theoretical foundation empirically, and we can utilize that theoretical foundation to do very many things. One of them is to actually construct the perfect samples, representative samples, so most of the experiments that we conduct are valid, and also they will be much more reproducible than what is possible today. So let's engage in this particular journey. And again, as I want to remind you that please do have a look at the Big Data Epidemiology presentation as this fills up. And I also mentioned sometimes some of the concepts, although the slides are there, I'm not delving too much into it to shorten the time that I take in this particular presentation. Here is an important point. The aim of a clinical trial to establish whether the treatment A is superior to the treatment B or non-inferior to the treatment B on average. It should facilitate sufficient information for the physicians to select a treatment for the next patient who walks into the clinic. So, the rendering of the information that is produced in a clinical trial to this effect is essential. Here are two other important points. One is about the effect induced by a treatment on patients. It certainly points out that it may not be distributed in a very symmetric, normal, smooth bell curve. And the second point is that when you synthesize evidence from multiple studies, usually such evidence synthesis would synthesize the mean or the average effects, and then the confidence interval describe the spread of the average effect. But it's very, very important to also know the different effects that are induced in different patients, so that the spread of the spread is also more important, and in some way of rendering that information to contain the spread is important. This slide and the next speaks about the creation of the coordinate system or the therapeutic landscape, or elsewhere I describe this as the basic theory of the epidemiology and the medicine. So with the big data, we have available to us many clinical efficacy and safety outcomes. We take them, there are many of them, and then using statistical methods, we reduce the dimensions and extract few dimensions of importance, and these few dimensions set up the coordinate system. I refer you to my presentation on big data epidemiology, which discusses this in detail. As I mentioned in the previous slide, this is discussed elsewhere, and also I picked this one up later in the presentation. You will see this in detail, and so you wouldn't miss anything. But I would still refer you to my other presentation of big data epidemiology. Again, this slide is here for the completeness. I refer you to my other presentation on big data epidemiology for an explanation. I will speak about this later in these slides. You won't miss too much, so I'll pause here. Here is a visualization which clarifies what we have discussed. This is a two-dimensional map. On the x-axis and y-axis are the reduced coordinates, and then the dots are individual patients, and then as I described and using the inferences, we have actually clustered these patients, and then these boundaries of these are the clusters of phenotypes. And I have made this point, and still this is only a two-dimensional projection of this coordinate system. In reality, it contains more coordinates, but even within the two-dimensional, you can still see that if we get closer, there could very well be the smaller clusters. Just to highlight this point, and you can actually see in some within the blue circles I've highlighted in gray some sub-phenotypes, if you want to call it, but it's in we need to remember that this is a population scale map, very large populations of even 7.5 billion, if you're lucky, patient populations. This visualization, in fact in animation, shows the multidimensional nature of this coordinate system. Here I've taken the three coordinates, first most important three coordinates, or maybe the first three coordinates of them, and then show the clusters. Actually, you see the patients themselves, and then on top of that I've highlighted in contours the clusters. And then with the animation, as it rotates, you can see the world, the islands, and the continents, and the patient distribution across these clusters. So this is just to show that it's a 3 dimensions, and if there are more than 3 dimensions, you can look at any 3 dimensions at a time in this way. Here is a visualization which clarifies what we have discussed. This is a 2-dimensional map. On the x-axis and y-axis are the reduced coordinates, and then the dots are individual patients, and then as I described and using the inferences, we have actually clustered these patients, and then these boundaries of these are the clusters of phenotypes. And I have made this point, and still this is only a 2-dimensional projection of this coordinate system. In reality, it contains more coordinates, but even within the 2-dimensional, you can still see that if we get closer, there could very well be the smaller clusters. Just to highlight this point, and you can actually see in some within the blue circles, I've highlighted in gray some sub-phenotypes, if you want to call it. But it's we need to remember that this is a population scale map, very large populations of even 7.5 billion, if you're lucky, patient populations. In big data and also in AI applied to the population scale data, visualization is extremely important component. As you are aware, we're working on extremely high dimensional data. And then in this particular case, we would have actually reduced the dimensions and went even a further step to show people the groupings of the patients by phenotyping them, creating phenotypes of similar patients. And as you are aware that these phenotypes have been constructed using the safety and clinical outcomes. These are very commonly measured in patients across the world in any particular disease area. And so we can actually project them back to these phenotypes. So here what you see is a series of phenotypes, and usually the number of phenotypes larger in the population scale data. Here I constructed 24 population scale phenotypes. Each of them is a radar plot, and each radar contains the almost like a bar plot in 360 degrees, and each particular arrow or each particular line shows the dimension of the variable, that original variable, clinical or safety variable. And these plots are also called butterfly plots because in general they tend to look like butterflies. But leaving aside, it does carry a very interesting message, and people, medics and others, can in general compare these phenotypes and show in particular set of variables they differ visually, and very easily and impactfully get an idea whether these phenotypes are different and in which way they are different in a level. Here is another projection or visualization of the therapeutic landscape map or the coordinate system. Here you see a two-dimensional plot, but in reality it is showing a three-dimensional effects. So on the x and y-axis are the coordinates that we have reduced, the reduction of the dimensionality. And then on the z-axis, which is depicted in here by the contours and the color shading, are showing us the effects of or therapeutic effects, I call it. And in fact, the concentric circles, each circle shows an outcome. The beauty of the circle, as you understand, is that you could actually put theoretically infinitely many sub-circles inside it. Therefore, here you see 1, 2, 3, 4, 5 outcomes, and then if there are 250, you could actually put a 250 outcomes, 250 circles, and each circle is showing the outcomes in a population of the whole. It's a very interesting outcome. I also have a three-dimensional view of it, but I didn't want to bring it in here. But I hope you get the point and also insights about the total population that we have modeled in the landscape. Okay, let's put them together. First, we collate all the variables, safety and efficacy outcomes from patients, clinical outcomes that are measured globally from the patients. And then we reduce the dimensions and extract few dimensions. Using that reduced dimensions, we further derive phenotypes if we opted to cluster them. And in this particular diagram, you see I show you two coordinates, first and the second, let's say, and then the dots are patients. And then you see these gray circles. They are clusters. Cluster boundaries has been established. And then we map the original clinical variables back into this landscape so that these clinical variables will explain for the physicians and others what these types are. And then we also map the molecular variables into this landscape, which I mentioned about something about endotypes. And you can actually see this, in this particular case, I'm mapping genes. And then you can obviously go further. You can categorize genes into pathways, and which gives you a pretty good understanding of what might be going on each phenotype. Some of them have better outcomes, some of them have poor outcomes, some of them have extremely bad outcomes. And that way you can determine which particular phenotypes are not serving better with existing therapies. So objective deriving the need for therapeutic development. And also it explains for the others which phenotypes are served better and treatments will work. And so this is the universal coordinate system and a 360-degree, if you want to call it, framework. And as I want to remind you once more that here I'm only showing the first two dimensions, but this is a multidimensional system. So in explaining the distribution of the coordinate system, as you remember, X and Y, we had the reduced dimensions, the coordinates, first and the second coordinates, and then here I take up the third coordinates and show the third dimension and make an animation to show the distribution of them. As you remember that these are multidimensional. Here we only see at three specific dimensions. And then I've actually projected the contributions from the original variables as a biplot. You remember that each particular arrow contains the original variable. The length of the arrow is proportional to the force that is applied. The direction is the direction that particular variable is driving the patient population. In one way of looking at it, all the patients are originally could be, assuming that they were normal of one way or the other, in the middle. And then as the outcomes differ in different diseases or the different stages of the disease, the patients get spread out and then form the group. And in the movie you can actually clearly see the inner three-dimensional in the third. So if you have more dimensions, we can look at three at a time. Another important thing that we can do when we have the coordinate system is that we can map the patients. In here the dots are patients, and as you remember, we only show the two main dimensions that we have reduced, and then we can cluster them to group the patients into phenotypes if we want. So here you see some circles drawn in gray, and then we can map both the original clinical variables that contributed to creation of these phenotypes and also the molecular variables that may also explain what I mentioned earlier called endotypes into the same coordinate system. And once you have this, it's one of the biggest assets we will have in a therapeutic area, especially for anybody who's trying to develop treatments for patients. Especially these days, we know when we actually think about a drug, we know the mechanism of the drug, the molecular mechanisms. Now, using the molecular variables that we already mapped onto this, we can actually map those treatment into this landscape. So as we see another plot, where here is where we are going to actually do the arithmetics in study design and all the stuff, but on top of that you can actually use this to compare different treatment that already exist or that are being developed. So we can use the map as some sort of an operational tool for doing the arithmetic on comparing these drugs as we continuing the development of them, bringing in those studies, the patient level data, performing the transformations as we have done into the coordinate system. That would allow us to do more precise comparisons, statistical comparisons between these groups. And then also, as I mentioned elsewhere, designing studies with high precision, assessing the effects of the drugs or the treatments in the patient population, segmenting in this way, that way, as we have discussed elsewhere. Just to illustrate the evidence synthesis process. So far we were talking about the studies or the patient level data, the arithmetic that we do to the patient level data, whether they're electronic health records, longitudinal cohorts, likewise. And here in the last stage, you just show the coordinate system, and then we derive the phenotypes, and then we discussed about the comparisons of these phenotypes. And at stage, we needed to map the existing studies, the clinical studies, often randomized controlled trials, into this landscape, and then compare the performance of them, because most of the time the patient level data for those are not available. And also for evidence synthesis, they are the gold standard, and any medic or any healthcare system or any medical society wanted to know how these novel new drugs compare to the existing in terms of the studies at a meta level, which is usually done in a meta-analysis. The art of meta-analysis is slightly different, and it's very well conquered. But here, this allows us to map them in the real world, the performance, patient level data. So what we can do here is that using the inclusion and exclusion criteria of those studies, we can map those studies into the phenotypes. And then here you see, usually this evidence synthesis is published as a forest plot. You see on the right hand side the forest plot, and each study in a particular clinical effect. It actually shows the confidence intervals as we discussed before. And then we see the studies, and we see them here more precisely mapped into the phenotypes. Now this allows us to compare those phenotypes, the effects of these treatments on different phenotypes, stratified way, which was not possible from the original clinical studies that are being published. That makes it the evidence synthesis much more perfect. So that can bring the evidence synthesis to the patient level, which is not possible in other scenarios. Without having the coordinate system and the data, we can't do this. So I hope this illustrates the point that the code system or the theory is helping us to integrate a variety of important data to make high precision evidence synthesis, where we can make therapeutic decisions applicable to each and every individual patient rather than just groups or average patient. I spoke about this in my presentation on big data epidemiology, so I'll refer you to that presentation. But it's worthwhile to have a look at of this particular slide because it actually explains in detail the operational environment that we can build around the coordinate system or the theoretical foundation that we have built. This actually gives us a framework for discovering new drugs for the evidence-based need and then taking the treatment development through the early clinical, late clinical, and then registration. And after that, when we go to the clinic, it also provides us with the framework to explain the benefit risk profiles in a patient-centric way rather than for an average patient. Now, it is also useful operationally in managing clinical development programs, and it converts them in an ad hoc activity as it is done today with a high failure rate to an engineering activity. So engineer therapeutic intervention for success. In the meantime, I also urge you to read the paragraph, the highlight in the conversation between Alice and the Cheshire Cat, which actually in a strange way describes what usually might happen in certain circumstances of developing a treatment in the industry today. Here's a thought. It's kind of an are we there yet kind of thought. It was a few years ago there was this concept of translating from bedside to bench. And then before that there was this bench to bedside. And then now we're just going in circles from bench to bedside and bedside to bench, and likewise over and over again. But here is a very interesting piece that I read from the article. You see the quotation, citation of it. It actually goes on to say, I will read this, and personalized medicine is often described as genomic-based knowledge that promises the ability to approach each patient as the biological individual that he or she is. This is an appealing description, yet unless clinical, social, and environmental features, which is in short big data, that affect the outcomes of disease are also incorporated, the current approach may be carving a path to depersonalized medicine, both in its science and its relevance to medical practice. A very thoughtful quote, and I hope you will reflect on it. Summary of the algorithms. First, we phenotype patients using available patient-level clinical observational real-world data. The outcome? Meaningful clinical phenotypes that can describe the potential therapeutic landscape is established. And then we map any available molecular expression data to the derived landscape. For example, gene expression, protein expression, any other mappable molecular expression data. As a result, we obtain a molecular understanding of the mechanisms involved in the strata which manifest these clinical manifestations. And then we map the current therapies, or it could be the existing therapies that are in development by us, means particular company or their competitors, via the molecular fingerprints. The outcome is the potential of the therapies in development can be established. Competition. And then we map the published summary level data, meta-analysis output to the current therapies of the current therapies or the therapies that are being investigated into the landscape. The outcome is what therapeutic effects are brought about by the potential therapeutic effects by these therapies to specific segments of the populations. Otherwise, I call it 360-degree or the total evidence synthesis can be established much better than the current meta-analysis. So in summary, the coordinate system or the therapeutic landscape map is a comprehensive synthesized evidence framework or a data reduction technique. It's a powerful tool for pinpointing the unmet need in stratified or precision medicine. It helps us to design new clinical development programs with high precision by fully characterizing the strata or the clusters in terms of their clinical characteristics, inclusion-exclusion criteria, and diagnostics. So, why I'm talking about this now? In early January this year, I have set up a charitable foundation and opted to call it Hospite Foundation. I hope you'll find what the meaning of hospite is. And it is also going to help patients to manage their own care. In a crowded space of healthcare systems, the most interest about the patient's health is patients themselves. So in some ways, the change of the scenery is necessary. Patients need to take the responsibilities for managing their own care. What is lacking today is the tools for them. Now, in the 21st century, most people are actually very fluent in manipulating electronic tools, apps if you want to call them. So if you create an app to manipulate their health, they will, not everybody, but majority of the people would be able to utilize them. And then that will also help actually the physicians, so that if we synthesize patient level views just before a physician sees the patient, it will help. And also for the healthcare systems, insurance companies, even for the pharmaceutical industry. So in essence, it is a kind of a buzzword, but you must bear with me, and I call it pivoting medicine back to the patients to manage their own care. And we want to change the world, and I hope you're going to join us one way or the other, sometimes by sponsoring our work. Sometimes I have a network of people who are extremely bright and smart and intelligent, working on the pieces of this, and together we can crack this. And I will have another sets of presentations outlining the missing parts of, or maybe explaining further some integrated parts, especially the AI-based framework that I'm building up, so that it will help individually, either you join us or not. And I hope this discussion, or the time that you spent in browsing through this YouTube presentation, is fruitful, and it will help you as a professional or an industry or a patient even. Thank you very much. Appreciate it.